Hello everyone. In this video, we are going to learn about carbonic anhydrase enzyme. So, the topic of today is carbonic anhydrase enzyme. In short, we call it CA. It is a very important enzyme present in our body, in our blood. It helps in the carry of CO2, that is carbon dioxide, from the tissues to the lungs. Now, carbonic anhydrase enzyme are found in various by organisms, and it differs from one organism to another organism, but their basic biological structure is same, and their biological activity is also same. So they are called isoenzymes, only are different in the outer structural part. Now, carbonic anhydrase is a zinc containing metallozyme. That is, the metal center is zinc, so it is a zinc containing metallozyme. Now, carbonic anhydrase, as we have said, that it helps in the CO2 transport in blood. So it is present in blood only. Now, what it does is it does the reversible hydration of CO2 in blood. CO2 by reversible hydration of CO2 in blood. Now, what do we mean by reversible hydration? We know that our tissues produce carbon dioxide by the cellular respiration or the glucose breakdown, and that carbon dioxide is carried in our bloodstream to expel out via lungs. So it is a gas and its solubility in blood is low. So in order to make it soluble in blood, in order to carry it to the lungs, we need this carbonic anhydrase enzyme, because the solubility of CO2 is very low in blood at our pH. I am going to discuss it after just a minute. Now, reversible hydration means it will hydrate the CO2 and it will dehydrate the CO2. Hydrate the CO2 to form bicarbonate ion and dehydrate from the blood to form CO2 again, which is expelled out via lungs. Now, I am going to write a series of equilibrium reactions at mentioning their rate constant at different pH. Then we will discuss and compare their rate constants. Now, first, CO2 is hydrated to form bicarbonate and H plus, its rate constant. Another series is CO2 plus OH minus to form bicarbonate, and third is again CO2 plus H2O in presence of carbonic anhydrase enzyme to form bicarbonate ion and H plus. Now let's write their pH, respective pH, and the rate constant. Here in the first reaction, the pH is that is the physiological pH, that is biological pH, that is pH of our blood. The second case is pH above 9.5, that is alkaline medium, and alkaline medium, then only we can get the source of OH minus, hydroxyl ion, and finally hydroxide ion, and finally it is also at biological pH, but in presence of carbonic anhydrase enzyme. Now let's write the rate constants. The rate constant in the first case is 10 to the power minus 1 second inverse, that is 10 molecules per second or 10 per second. Okay, now the next is 10 to the power 4 mole inverse second inverse, and the third is K equal to 10 to the power 6 second inverse. Now see, let's compare now. We have written the three equations. See, first, all these are hydration equations, and in the first case, the hydration at biological pH of CO2 in water is the rate constant is very low, so the rate is very slow. Number 2 is, second reaction is, in presence of hydroxide ion, that is in alkaline medium, this hydrolysis reaction is very high, that is 10 to the power 4. But the problem is that the biological pH is around 7 to 7.2 or 7.4, which is almost a neutral medium. So we need strongly basic medium, alkaline medium, in order to get hydroxide ion. But that is not possible for our biology. That is not possible in blood. But in biological pH, the same hydration reaction in presence of water molecule, in presence of carbonic anhydrase enzyme, the rate constant becomes very high, that is 10 to the power 6, which is really high. So that means the activity of carbonic anhydrase is very good, and in presence of carbonic anhydrase, a lot of hydration of CO2 molecule is possible in our biological pH. Now this is called the turnover number. That is, as we know, what is the turnover number? It is the number of substrate molecule which is transferred or which is transferred by the enzyme. In this case, the number of CO2 molecules which is transferred in the blood are in the form of bicarbonate. Hydration of CO2 occurring per molecule of enzyme when the enzyme is fully saturated with the substrate. So that is called the turnover number, per unit time per molecule of enzyme. How many substrate are converted into bicarbonates, and that turnover number is very high for carbonic anhydrase. And carbonic anhydrase has some more application. What is the number 1 is hydration of carbonyl compounds, and number 2 is ester hydrolysis. So they find various other applications in our biological medium as well. Now we will talk about the structural feature of carbonic anhydrase. Before that, we have already discussed that we can find many isoenzymes in isoenzyme means they are structurally similar, functionally similar, but they are of different kinds. Their functions are similar, but one might be slower, one might be faster, or some might be a bit only a small portion is structurally different, but overall basic structure or the framework is same. Based on their functions, based on their activity, carbonic anhydrase is structural feature are of three types. Structural feature, they are of three types. One is carbonic anhydrase 1, number 2 is carbonic anhydrase 2, and number 3 is Ca3. Now Ca1, among these three, Ca2 is the high activity form, that is their activity or turnover number is the highest. So high activity form, its turnover number is 10 to the power 6 per second. Carbonic anhydrase 1 is low activity form, that is its turnover number is 10 to the power 5 per second. And the carbonic anhydrase 3, it is very low activity form. Very low activity form, and its turnover number is 10 to the power 3 per second. and the carbonic anhydrase found in human, that is the human source, they are termed as HCA1 and HCA2. The 1 and 2 has the same meaning here also. Now, in all isoenzymes which are structurally similar, the molecular weight are approximately 30 kilodalton. Dalton is a weight measurement for the molecules or proteins and all. Now, each of the isoenzyme has a zinc center, Zn2 plus center per molecule, and the zinc acts as the prosthetic group, that is, without it the functioning of the apoenzyme, that is the other structural part of the enzyme, is null, that is, it won't be active, it is completely inactive without the zinc metal center. Now, this zinc is present inside a single chain polypeptide, that is its center, its pocket is made by a single chain polypeptide. Now, since HCA2, we will now go into the details of human carbonic anhydrase 2, which is the most active form or the high active form of carbonic anhydrase found in the humans. In HCA2, Zn2, that is the metal center, has a distorted tetrahedral geometry. We know that it is coordinated, it forms a coordination complex with the enzyme, making the enzyme, so it is distorted tetrahedral shape or geometry. And since tetrahedral, so its coordination number is 4. The 3 of them is satisfied, the 4 coordination number. The first one is histidine 94, second coordination is histidine 96, the third coordination is histidine 119, and the fourth one is either water molecule or OH. This is the fourth coordinated site, and which is the most important site. We will talk about this when we will the importance of H2 and OH, which acts as the nucleophile. I will talk about this when we study about the catalytic cycle. Now, the active pocket size, where the CO2 will be hydrated, has ordered network of water molecule or various amino acids. So we can write that the active pocket site has ordered network of water or amino acids, such as amino acids which are important for hydrogen bonding. For what? Hydrogen bonding, proton transfer, and hydrogen bonding is to stabilize the structure, and proton transfer is the RDS, rate determining step. While we study about the again in the catalytic cycle, everything will be clear. It is the RDS or the rate determining step, or the rate determining step is that step which is the slowest in the smaller steps of the entire substeps of the multi-step reaction, that is this catalytic cycle. So the order of the reaction, or we can say the rate of the reaction, depends on this step only. Okay, and and the amino acids that are present are either threonine 99, 199, and glutamate 106, or histidine 64. they play very important role in the proton transfer, and which is the RDS of the entire catalytic cycle. Now let's look into the structure of carbonic anhydrase. We can see that the metal center is here. The three coordination out of the four are coordinated by histidine 96, 94 and 109, and the fourth one is the OH group, and that is hydrogen bonded by threonine, the amino acids glutamate, and we can see there are series of hydrogen bonding here. So we can see that it is a tetrahedral geometry. It is forming tetrahedral geometry. You see, a distorted tetrahedral geometry like that, and there is this distorted means we can see that the zinc and oxygen bond is longer. That is Jahn-Teller distortion, elongation is here, and so these are the coordination number of zinc is 4. So this is the basic structure. So histidine, there can be histidine 64 as well, and series of hydrogen bonding we can see. So the main center of action is here. Now as we can see that the prosthetic group of this enzyme is the zinc 2 plus, without which the zinc enzyme is completely inactive. Now we have checked this by removing the zinc metal center from the prosthetic group to get the apoenzyme, the only apoenzyme without the zinc. Now this removal of zinc from this enzyme of carbonic anhydrase was done by the dialysis of the enzyme with a very strong chelating agent. We know that it forms cyclic coordination, that is more polydentate ligand or multidentate ligand, having, we know, multidentate ligand with two or more metal donating electron pair donating center or the ligand center. They are coordinating with the single metal center to form a cyclic structure like this. This chelation can be done. This is a chelating ligand, ethylene diamine. They can chelate with their electron pairs to a metal center, and 5-membered ring is formed. So this is a ring formation, that is chelation. So prosthetic group zinc can be removed from the, it can be removed from the carbonic anhydrase by dialysis with a very strong chelating ligand. Another example is EDTA, and here it is done by phenanthrolene. Okay, so this is all about the structure of carbonic anhydrase enzyme. In the next video we will discuss about the catalytic cycle of carbonic anhydrase enzyme, that is HCA2. Thank you.